Hello, my name is Nomaria Sharina. In this video, I will present my case study titled Aerobic Respiration and Fermentation. So this is the equation for cell respiration. One molecule of glucose, that is C6H12O6, and 6 molecules of oxygen, that is 6O2, and are converted into 6 molecules of carbon dioxide, that is 6CO2 plus 6 molecule of water, 6H2O. So next I will explain about stage of aerobic respiration. So apparently there are 4 steps of aerobic respiration. The first is glycolysis, that is break down glucose into 2 molecules of pyruvate. Second is link reaction, that's pyruvate convert into acetyl coenzyme A. The third stage is Krebs cycle, or also known as citric acid cycle. It's complete the breakdown of glucose. And the last is oxidative phosphorylation. And the fourth is oxidative phosphorylation, account for the most of the ATP synthesis. So the first is I will explain about glycolysis. The first is phosphate from the hydrolysis of molecule of ATP is added to glucose, a 6 carbon sugar, to form glucose 6-phosphate. Second is the glucose 6-phosphate molecule is rearranged into an isomer called fructose 6-phosphate. And third is second phosphate provided by the hydrolysis of the second molecule of ATP is added to the fructose 6-phosphate to form fructose 1. And then the 6 carbon fructose 1,6 biphosphate is split into 2 molecules of glyceraldehyde, 3-phosphate, and 3 carbon molecule. Then oxidation and phosphorylation of each glyceraldehyde 3-phosphate produce 1,3-biphosphoglycerate with a high energy phosphate bond and NADH. Then through substance level phosphorylation, the high energy phosphate is removed from each 1,3-biphosphoglycerate and transferred into ADP, forming ATP and 3-phosphoglycerate. Each of the 3-phosphoglycerate is oxidized to form a molecule of phosphoenol pyruvate with a high phosphate bond. Through the substance level of phosphorylation, the high energy phosphate is removed from each phosphoenol pyruvate and transferred to ADP, forming ATP pyruvate. So that is for glycolysis. Next is link reaction. So link reaction is the process of pyruvates to convert into acetyl coenzyme A before entering Krebs cycle. Connect glycolysis to Krebs cycle through a process called oxidative decarboxylation. So this is the Krebs cycle, and there are 8 steps inside the Krebs cycle. So the first step is the citric acid cycle begins when coenzyme A transfers its 2-carbon acetyl group to the 4-carbon compound oxaloacetate to form the 6-carbon molecule citrate. This is the first step. And then second step, the citrate is rearranged to form an isometric form, isocitrate. This is the isocitrate. And then next is the 6-carbon isocitrate is oxidized, and a molecule of a carbon dioxide is removed to produce the 5-carbon molecule alpha ketoglutarate. And during this oxidation, NAD+ is reduced and During this oxidation, NAD+ is reduced to NADH and H. The second step is alpha ketoglutarate is oxidized, carbon dioxide is removed, and the coenzyme A is added to form the 4-carbon compound sulfonyl coenzyme A. During this oxidation, NAD+ is reduced to NADH and H. Okay, the fifth step is coenzyme A is removed from the succinyl coenzyme A to produce succinate. Here. The energy release is used to make the guanosine 3-phosphate, also known as GTP, from guanosine disphosphate, it is GDP, N-pi by phosphate level phosphorylation. GTP can then be used to make ATP. The sixth step is succinate is oxidized to phenolate. During this oxidation, FAD is reduced to FADH2. The seventh is water is added to phenolate to form malate. And the last step is malate is oxidized to produce oxaloacetate, the starting compound of the citric acid cycle. During this oxidation, NAD plus is reduced to NADH and H. So the next stage is oxidative phosphorylation. Oxidative phosphorylation generates the majority of ATP in aerobic respiration. It involves electron transport chain, chemiosmosis, and ATP synthesis. Okay, so apparently there are major difference between aerobic respiration and anaerobic respiration. The first difference is in aerobic respiration, in this respiration, it uses oxygen to break down the glucose. Meanwhile, in anaerobic respiration, it doesn't use the oxygen to break down the glucose. Next, in aerobic respiration, carbon and water are at the end of the product. Like this, for example, carbon and water are in the end of the product. However, in anaerobic respiration, alcohol or lactate are at the end of the product. Also, in aerobic respiration, it can be found in the cytoplasm and mitochondria. However, in anaerobic respiration, it can only be found in cytoplasm. There's no anaerobic respiration in mitochondria, only cytoplasm. Okay, so next I will explain what are the significance of NAD plus in fermentation and cellular respiration. So the first is the NAD plus molecule is a shuttler of the electron and hydrogen atoms. In fermentation reaction, NAD plus molecule pull electrons atom of nutrient molecule, for example sugars, and transfer them to pyruvate molecules, forming fermentative end product like lactate or ethanol. Type of end product depend on the type of the cell that is fermenting. In cellular respiration reactions, NAD plus molecules pull electrons atom of nutrient molecules, for example sugar or fats, and transfer them to electron transport chains. This is electron transport chains. Okay, so the next question is, why do organisms undergo fermentation in absence of oxygen? So, when oxygen is not present, or if an organism is not able to undergo aerobic respiration, pyruvate will undergo a process called fermentation. Fermentation does not require oxygen, and therefore it is called as anaerobic respiration. Fermentation will replenish NAD plus from the NADH and H to produce in glycolysis. Okay, for example, this usually happens during workout or exercise. In the beginning of our muscle undergo the cellular respiration. However, as we keep exercise for too long, our muscle will eventually lacking of oxygen. Therefore, it will undergo fermentation respiration to produce lactic acid to provide more energy to our muscle. So, in conclusion, both aerobic and anaerobic respiration are really important in our life. In cellular respiration, it provides the energy for living organisms to perform all of the other necessary functions to maintain life. Meanwhile, as for fermentation respiration, it is used when human muscle cells cannot gain enough oxygen to undergo cellular respiration. Thus, these two respirations are related to each other as it provides energy to us. Not only just for human, but these are also important in other different aspects. For example, fermentation process is used in production of alcohol beverage, bread making, and some other food like cheese, tempeh, or yogurt. Okay, here is my list of reference. There are total of 5 references that I used in my presentation. Okay, so that's all from me. Thank you very much for watching this video.